Hey, welcome back. This is Delon with Paying Attention, where I demystify the science behind bodybuilding and bridge the gap between conventional fit wisdom through my own experience. Today I'm going to be talking about the glass ceiling of muscle composition in bodybuilding and how to hack your own biology, or a couple ways that I'm going to go about doing it, and I'll share some of my own experiences from the information that I've gathered thus far. As far as myostatin and inhibition is concerned, its main role is to make sure that your myoblasts don't build more muscle. I am really uncertain as to why that's even a thing, but it is, and it's a hormone that does that. Myostatin outside of the bodybuilding community is an aging population concern and is usually brought up when people experience fatigue or muscle dystrophy in their twilight years, or as they get older. The gene sequence was discovered in the 90s during some gonadal in vitro research and has been regulated to clinical trials for muscular dystrophy and sarcopenia ever since then. There's one study I read where it said that, I think it was from Muscle and Fitness, where they said that there's like a direct correlation between progressive overload and how and your myostatin levels, and what they indicated was that, maybe I can find it and I'll post it up here, but the more that your body has to respond to uh, challenging workouts, the more myostatin is endogenously or endocrinally inhibited, so you can naturally suppress it on your own through heavy weightlifting or challenging exercise. Uh, but that's not always enough for some people. In bodybuilding, myostatin and activin are antagonists to muscle proliferation via myoblast or sarco and hypertrophic packing. As I mentioned in a previous video that I'll tag up on the top, oftentimes elite bodybuilders who are genetically labeled as gifted guard their efforts and hard work. Um, Not always, but sometimes are blessed with a polymorphism in this gene expression. Um, the gene sequence is RS180508-6. The polymorphism that I was just talking about is K153 ARG-R, or K153R for short. Normally there's only two alleles, and the two alleles are C and T. CC, if you have the CC allele, you are in the upper echelon. And then under that would be the CT, which is a combination of the C and the T alleles. And then the very bottom of the, which has the most myostatin inhibition, would be TT. People with this polymorphism, which is less than 0.0001% of the population, don't have any myostatin inhibition. And there was one study in 2004, and I'll post a link in the description below, for it was like a, I don't know if it was like a toddler or it was a young kid, and he had this polymorphism, and the doctors were essentially baffled by this kid's extraordinary muscle development. And they even called it precocious. And yeah, it was his physical this is what it reads right here. His physical prowess was precocious along with his development, and he had this KR153R polymorphism. People that have this polymorphism, it lets them pack on muscle in like a super physiological sense in response to caloric surpluses and exogenous anabolic steroids. The polymorphism inhibits myostatin, or directly just doesn't make it, which is myostatin, as I talked about earlier, is a protein secreted in muscle tissues known as GDF8 or growth differential factor 8. Another protein similar that inhibits muscle growth is activin type 2, R&B, I believe. I'll correct myself if I'm wrong down below. But both of these proteins bind to myoblasts and inhibit muscle proliferation. So myostatin was discovered in the 90s, I said earlier, because they were doing gonadal and vitro research. And it's unfortunate for people that have muscular dystrophy because this drug inhibition has been stuck in clinical trials for almost 15 years. They also found in a couple lab rat experiments that not only does inhibiting myostatin increase your muscle proliferation, but it also increases the tendon strength and your bone strength as well through BMD, which is bone modifier density, I believe. And so inhibiting these is turns out is actually already a factor of your endocrine system, but you can supplement that, which is what I intend to do. Follistatin is directly stimulated by your gonads through your pituitary glands, and then what it does is it binds to your proteins, and then it uses autokernel processes so that they inhibit myostatin or activin type 2 from binding to these myoblasts, which proliferate muscular growth. So essentially by introducing exogenous FSH or follistatin hormone, you're increasing the bioavailability of your myoblasts' ability to regenerate new muscle. FSH is endogenous and regulates ovarian and reproductive processes, but also has 3 isoforms: FSH 315, 288, and then there's a follistatin-like hormone. Can't remember the name at this particular moment. But of the 2 isoform, or the 2 that I was researching, 288 has autokernel inhibiting factors, but 315, which is a synthetic peptide, they're both. The one that I ended up purchasing was a synthetic peptide, but I'll get to that in a little bit. 315 doesn't have the heparin protease binding affinity that 288 does, but when it actually enters the cell, is transformed through the membrane to resist activin, essentially. So it goes from a weak antagonist to activin to something that is equally as antagonistic towards activin binding and GDF8 as well as FST 288 does. and it biosynthesizes a lot faster as long and secretes a lot faster, so it's faster acting than FST 288. So that is the one I ultimately ended up going with from one of those peptide uh, online websites. I'll post down below if you're interested to check it out for yourself. Thanks again for tuning in to paying attention. Don't forget to like and subscribe, and let me know in the comments down below if you have any experience with FSH or follistatin and exogenous hormones, and I'll see you in the next video.